No.
Well, it's posted as an assignment. And I've written a description there. It turns out that the biology department is doing a survey looking at what's called student learning outcomes. And they've chosen the topic cell division. So they're asking each section in Bio 130 to respond to this survey to see how well, how much retention there is on a particular topic. And as I said, they seem to have focused in on cell division. Well, we've gone over that already. And so that's why I submitted it as an assignment. You'll get credit for it. And it's open book. So you can use any resource that's available to you to answer the questions. So I strongly encourage you to do that. You get extra credit for it, and it will help the department achieve its goals. So that's point one. Point two is that the worksheet on enzymes and catalysis is due tomorrow. And then we'll discuss it on Wednesday in class. And I would like to now share my screen with you. And go over the quiz 7, that we did last week. The average grade was 73%. Several people got 100% on this quiz. And I'd like to go over and emphasize some of the deficiencies. Obviously, some of you didn't have any deficiencies, but some of you did. If you look, can you all see what I'm looking at here? The quiz.
Yeah, I can see it.
Quiz 7, and can you read it? I'll make it a little bit bigger. Okay. First question was, the goal of mitosis is, and second part, the goal of meiosis is. Well, what we're looking for there is, goal of mitosis is exact reproduction, and the goal of meiosis is variation. Those are the main features that we tried to emphasize in going over these two processes. About 9% got it exactly right. Most people got partial credit because we gave credit if you didn't say something wrong. But most people who responded to this question gave information about mitosis and meiosis, but didn't hit the nail on the head. So if you got partial credit on it, that's why. And I just want to emphasize the parts of the slide set that this is relevant to. One of the slides on mitosis says the consequences are two cells identical to the progenitor, no genetic variation. And on meiosis, the genetic variation is created. You start with a diploid and end with a haploid. But the main thing I want to emphasize here is that there's a summary slide in the set comparing mitosis and meiosis. And right at the top in red, the goal of mitosis is exact reproduction. Goal of meiosis is to introduce variation. So that's why that question was there, and that's why it was graded as it was. All right, the second question is about genetics, Mendelian genetics. About half of you got it right. You had to choose among several possible answers, and about half of you chose 50%. The other half didn't, and so that's what I want to focus on. Why didn't you? So if you read the question, hemophilia is an X-linked disease. What is the likelihood of a male with hemophilia passing this trait along to his progeny? Okay, but what about the female? The female's condition is not given. So, since it's not given, she's not a hemophiliac, and therefore she's normal. So her genotype then will be big X, big X. Well, the male is a hemophiliac, and since this is X-linked, there's only one possibility: his X chromosome is mutant. So his genotype is X star Y. So these are the gametes. A male can produce an X star gamete or a Y. Female can only produce X gametes. And so if you do a Punnett square, you can see that the females, the ones that are XX, will all be carriers, and the males will all be normal. And the females are carriers because in order to be a hemophiliac, a female needs to have a mutation on both of her X chromosomes. So that's how we come up with the conclusion. The answer is 50%. Is that clear? The thought process you go through to come up with the genotypes of the gametes, and then all the possible combinations of fertilized eggs. Okay. The next question had 5 parts, and the point of this question was to ask if you know the various phases of meiosis and what happens in some of them. So crossing over occurs at prophase of meiosis 1, whereas random assortment occurs at metaphase of both meiosis 1 and meiosis 2. The information that you were presented about this is shown in the slide on meiosis 1. Prophase 1, crossing over occurs. Genetic variation was emphasized here. And then in the very next phase, metaphase, independent assortment occurs. Again, genetic variation. We emphasized the crossing over part of it, where the chromosomes are in intimate contact. They form these tetrads and exchange parts. That's where the variation comes in prophase. Then if you look at independent assortment, it happens in both metaphase 1 and metaphase 2. Again, the point here, it's a very important source of genetic variation because at both of these phases, the chromosomes randomly assort to either one side of the cell or the other. About 10% of the class got all 5 parts right. Most of you missed at least one. And question number 4, match the description with the most correct term. The gene that contains 2 identical copies of an allele is homozygous at that locus. This next part what took a little thought. Someone who was a carrier of a disease condition, you had to think about that, but you were limited to the choices on the list, and the only choice that fits is heterozygous. 1 copy of an allele masks the phenotype of the other copy. That's complete dominance. A set of alleles that discovers the expression of a protein is a gene. Crossing individuals produce a progeny with an intermediate or blended phenotype. That's incomplete dominant. And a relaxed form of DNA is chromatin. Number 5, in the cell cycle, mitosis occurs in the M phase. Almost everyone got that right. And finally, there's this essay question. It's another Mendelian inheritance question. So you're growing a flowering plant that has 2 possible colors, orange and blue. Now the question states that orange are recessive. That means blue is dominant. So you have spent some time producing true breeding plants. That means you inbred them so that they were all homozygous at all loci. Both types of plant, both colors were homozygous at all loci. And then you do a cross an orange and a blue. And the question asks for phenotypes. What phenotype do you see in the F1 and what phenotype in the F2? So this is a simple monohybrid cross. But only about 10% of you got both parts correct. About 70% got partially correct. So what you have to look at is, first of all, that orange is recessive, and that you've spent some time making inbreds, true breeding inbreds, homozygous at all loci. So since orange is recessive, and if we give the locus the symbol B, then orange is going to have a genotype of little b, little b. Blue is dominant, so its genotype will be big B, big B. The parental cross is between a big B, and a little b, little b. So the F1 is all heterozygous, and the phenotype will be all blue. The F2 is a cross of 2 F1s. So since all the F1s are heterozygotes, it's a cross of 1 heterozygote with an identical heterozygote. And if you do the little Punnett square, you can see that you get 1/4 homozygous dominant, half are heterozygous, and 1/4 are homozygous recessive. So that means, to answer the question, in the F2, the phenotypes are 75% blue and 25% orange. So I thought that this would be fairly easy, but it wasn't for most of you. And I want to encourage you, if you're having trouble with these inheritance questions, figure them out. Go to Scott's workshop or study the slides, do something to figure out how to do them, because they will come up again. We're going to have a test 3, a week from today. There will be questions of this type on that test. So you need to know how to do a simple Mendelian inheritance.
Professor, if I may throw something in there, don't forget you guys have a discussion board thread where you can ask me questions, and you guys have a Discord where you can ask each other questions. And I also keep an eye on the Discord, and if I see something, I would answer it there. So if workshop doesn't work for you, we have provided one alternative through school, that being Canvas, and you guys have provided your own alternatives with the Discord. So use those resources. Don't just let this stuff fester and you don't get the right answer, because if you work a couple of these, you should have no problems. They're fairly simple type problems. It just takes a little bit of practice is all. And sometimes you just need that little, somebody points out one thing and you go, oh, that was it. Got it.
Yeah, and to emphasize, you need to read the questions carefully and see what information you can get out of the question and the way it's asked. So for example, if we go back to, I think it was question 2, yeah, the hemophilia one, at first you say there's something missing here. There's nothing is said in the question about the female. Well, that's where you think about it and say, if the female was a carrier or had hemophilia, it would say so. And since it didn't, you assume she's healthy, normal. All right, anything else about the quiz and thinking about the test coming up in a week? We'll stop.
Professor, for the quiz, do we have to attempt?
Excuse me, for the test?
Yeah, do we have to attempt or it's one?
Oh, one attempt, yeah. The test will be a full period, 75 minutes, and it will be worth 100 points.
Okay, there's no chance to be to attempt?
No, this test will be one attempt.
Okay, thank you.
All right, we're back to where we were last time, talking about energy and enzymes. And again, I want to remind you of the overview of where we're going in this next section of the course. We're talking about energy. What is it? Where does it come from? Enzymes, their structure and function, mechanism of action, and then how both of these relate to two fundamental processes in biology. One is photosynthesis and the other is respiration, which are the reciprocal of each other. So I'm just going to touch base on a few points I want to emphasize from last time. And then we didn't finish this slide set last time, so today we're going to finish it.
Just a quick question. Did you post this?
No, I haven't yet because I've been continuing to revise it, but I will post it after class today.
So, because we have assignment and there is a lot of question from this lecture.
Right, and that assignment is due tomorrow. So I will post it and you'll have access to that to complete the worksheet.
Sounds good. Thank you.
I want to emphasize a point here about the electromagnetic spectrum. We went over this last time, and you remember that there's only this very small little clear section of the spectrum that we humans can see, and that in fact plants can see. It's called visible light. And plants can absorb reds and yellows and blues. They cannot absorb green. So they reflect it. And that's why when you look at a plant, it appears to your eye to be green. It's reflecting green light. But the point I want to make now is that when you look at plants, you'll see it's pretty obvious that not all plants are green. So what are the possible reasons why a plant might not be green? Well, the first possibility is that it is really green, it has chlorophyll, which is masked by another pigment. And an example of that is shown here on the right. A red maple is red. If you look at the other green plants here, red maple shows no sign of being green. But it's got chlorophyll, but the chlorophyll is masked by a red pigment, one of the anthocyanins. So that's one reason. If you see a red tree, it's probably an ornamental tree that overproduces anthocyanins. Well, another reason a tree or a plant might be yellow is that it's nutrient deficient. And very often when a plant isn't getting proper nutrition, it will turn yellow. So it's got chlorophyll, but it may be degrading it because it's not getting proper nutrition. Another possibility is albinism. It could be genetic. So for example, here are corn seedlings. One is normal green, the other is pure white. And that's because it had a single gene mutation for a protein involved in photosynthesis. So it simply cannot make chlorophyll, so it's white, not green. Now, interestingly, this little seedling is growing on the nutrients present in the seed. As soon as those nutrients are exhausted, this little plant will die, unless you feed it sugar. If you give it glucose or sucrose, for example, it will continue to grow. It'll never be green, but it can grow. And finally, if you see a tree that isn't green, or any plant that isn't green, you may ask, what time of year is it? If it's in the fall, it might be that it's shedding its leaves. It's a deciduous plant, and its leaves are falling. So here's an oak tree in the autumn, and you can see that it's showing no sign of green. Many of its leaves are already on the ground. They've fallen off. So that is yet another reason why you might see a tree that's not green. And in fact, this tree at one point was green, but it receives signals in the fall that cause it to degrade its chlorophyll. So chlorophyll is actually gone in a tree like this. And finally, a plant may not be green because it's a parasite. Well, this picture on the upper left here is actually one taken by Scott. He was driving out in the desert. What you see is a green plant with a yellow patch on it. This yellow patch is also a plant, but it's not green because you see here up close, it's a parasite. It's feeding off the green plant. So the green plant can make food from photosynthesis. The parasitic plant taps into that food. And I've shown here one of the ways that a parasitic plant can do that. It makes these little tendrils, little structures that actually penetrate into the host plant. These are called haustoria. And those are the feeding structures. And you see a micrograph, a cross section of a host parasite interaction here. Parasite is on top, the host is on the bottom, and you can see the little structures called haustoria, which are feeding organs. They suck nutrients out of the plant, primarily sugar, but the parasite gets its entire nutrition from the host. And in fact, this is not a healthy relationship for the host. If the parasite is growing on a plant long enough, it will usually kill it. So to go back, I want to emphasize that plants tend to be green, and if they're not, there are quite a number of reasons why they might not be. I want to page ahead. To here. And review what we went over last time, talking about endergonic reactions and exergonic ones. Endergonic require net energy input. Exergonic reactions release energy. And to illustrate that, we contrasted photosynthesis with respiration. In the case of photosynthesis, the process requires energy. That energy comes from sunlight. Requires energy to take carbon dioxide and water and make glucose. Respiration is the opposite. It starts with glucose plus oxygen, breaks glucose down, and as it does, energy is generated. So examples of endergonic and exergonic reactions. And I want to emphasize another point that we went over last time, and that is, how is activation energy lowered by an enzyme? Remember, we talked about activation energy in the case of sucrose and sucrase. The activation energy is very high. Without the enzyme, it's much lower. With the enzyme, but the net energy is the same. So that's the activation energy. And then how does the enzyme actually do it? I think this slide illustrates that as well as can be done at a superficial level. If you have the green here is the substrate, the yellow is the enzyme. There's an active site where the substrate fits into that. And when it does, the enzyme changes shape. And when it does that, it breaks or weakens the bonds in the substrate and it falls apart. And remember, the bonds that hold the enzyme together are multiple, and it's something to keep in mind when we talk about enzyme activity. I emphasized last time that you should know what a reaction rate means. It means the rate at which a substrate is converted to a product. So the speed of that transformation is the reaction rate. And here it's plotted against concentration of substrate with and without enzyme. And I want to re-emphasize denaturation. Enzymes can be damaged either permanently or temporarily by extremes in their environment. Extremes of temperature or pH or salt. And this is called denaturation. That's illustrated here at the bottom. Normal protein on the left. So its tertiary structure. Tertiary structure is required for activity. And then in the presence of an extreme environment, the bonds begin to break in this tertiary structure. And you end up with nothing but the primary structure, just a polypeptide. Amino acids joined together by covalent peptide bonds. And in this form, the protein has no enzymatic activity at all. In some cases, if you reduce the offending environment, say readjust the pH, the denatured protein may be able to renature again and regain its activity, but not usually. And then this illustrates a couple of those points. At the top of this graph is the effect of temperature. So the temperature of the human body is 37 degrees. And if you look at most enzymes in a human body, they have a temperature optimum of 37. So that is, if you look at the whole curve, at 0 degrees, enzyme would have no activity. And as you increase the temperature, activity increases up to a point. At 37, you go above that and getting warmer and warmer, and the enzyme starts to denature. So by the time you get to 50 degrees centigrade, it's completely come apart, has no activity at all. Now, not all enzymes are like that. So in heat-tolerant bacteria, for example, there's this one case in particular, the optimum temperature is 75 degrees, temperature at which enzymes, all enzymes in humans would be totally inactive. This is its optimum. And it starts to denature above that, but it can tolerate up to 95 degrees. So that if you then lower the temperature from 95 to 75, it's still active. Now with pH, we looked at 2 examples. This is, again, is in humans. So the optimum pH for pepsin is 2. The pH in the stomach, where pepsin is found, is 2. Whereas if you go farther down the digestive tract to the pancreas and the duodenum, the pH there is very close to neutral, 6 to 7. And the optimal pH for trypsin, which is an intestinal protein, is about 8, between 7 and 8. So it depends on the part of the body that you're in as to what the pH optimum for enzymes in that organ would be. The stomach is an unusual organ. It has such an extreme pH. Have any idea why? Why would a stomach have such a low pH? It's almost like pure acid.
To assist in digestion?
Yeah, that's one possibility. And in fact, that is true. But it does so by activating enzymes that are active at low pH. So yeah, assisting in digestion is one thing. But think about the food you eat. Do you sterilize it before you eat it?
Yes, of course. Doesn't everybody? Everybody's got a UV machine at home, duh.
Yeah, so what does that mean? You're eating septic food.
You gotta, the acid basically kills any pathogens.
Right. And that is a main function of this extreme acidity in the stomach. And then I want to go over with you again the ATP, ADP interconversion, because that is so important in energy metabolism. And this diagram shows adenosine and AMP. If you take adenosine and add 1 phosphate to it, it's adenosine monophosphate. 2 phosphates, it's ADP. And 3 phosphates, it's ATP. And it's this last bond between the phosphates 2 and 3 that is high energy. So it takes a lot of energy to form that bond And when that bond breaks, it releases a lot of energy. So you can think of ADP, ATP is like a toggle switch. And this diagram illustrates that. If you start with ADP plus phosphate and you add energy, you can make ATP. And we'll talk about where this energy comes from. And when energy is needed, this bond is broken, so energy comes out, and you're back to ADP and phosphate. And this coupled reaction goes on constantly in most of our cells because we're constantly requiring energy, and we're constantly requiring a source of energy. We eat food to get energy to keep this cycle going. And then I want to again emphasize the two types of pathways, anabolic and catabolic. Anabolic pathways build up molecules and require energy to do so. Require free energy from the environment in order to take small molecules and make large ones. Catabolic pathways are the opposite. They tear down. They start with big complex molecules, usually polymers, and form simple ones. And in the process of doing that, as they break bonds, energy is released. And this illustrates anabolic and catabolic. So you can think of monomers like, for example, sugars like glucose or sucrose. Plus energy, you can make a carbohydrate, a polymer. A catabolic pathway just reverses that. You take a polymer like a carbohydrate or a lipid, and you break it down into monomers and release energy in the process. You can visualize catabolic pathways as in terms of the food you eat. If you think about what is in the food you eat, it's largely proteins, polysaccharides, and lipids, all complex polymers. Well, a catabolic pathway breaks these substances down through many different steps to what's called a citric acid cycle, where you actually get ATP. And these can be reversed in an anabolic pathway, so you can go from the TCA cycle back to complex molecules by interjecting energy. So these are anabolic pathways, and these are catabolic. Photosynthesis and respiration. We're going to go into each of these processes in detail in future classes. But the point to be made here is that one is anabolic, photosynthesis, where you go from carbon dioxide and water plus energy to make sugar and oxygen. And the catabolic process is respiration. You go from glucose and oxygen to make carbon dioxide and water, and energy is released. And now I want to emphasize, this is, I think, where we ended last time. And so from here on, it's, I think, new information for you. Pathway regulation. Why are we emphasizing this? It's because enzyme activity has to be regulated. If enzymes ran full speed 100% of the time, the cell would be destroyed. So when you need enzyme activity, you want it for the period that it's needed, but once it's no longer needed, it has to have a way of getting shut down. So we have enzyme inhibition, which can be irreversible or reversible. And it can be competitive or non-competitive. So these are reversible enzyme inhibitions. In this case, it's competitive. So you see the enzyme with its active site. There are two possible items that can insert into that active site, either the substrate or a competitive inhibitor, which is shaped just like the substrate, but it's not. It's not the substrate. So if this inhibitor is occupying this active site, there's no room for the substrate and there's no reaction. So that's competitive inhibition. Now, non-competitive is illustrated in the other diagram. Again, you have same enzyme, same substrate. This substrate will fit into this active site unless the non-competitive inhibitor inserts at a different site. So if the inhibitor is here, what it does is change the shape of the enzyme. So now the enzyme active site will no longer fit the substrate and there's no reaction. So that is a non-competitive inhibition of an enzyme. Clear to everybody? Make sure that you understand all this. It will come up. Now this illustrates what I think is the coolest way to regulate an enzyme. It's called feedback inhibition. So you have a substrate here in blue box, and it's the start of a pathway. Goes through another blue box, enzyme 2, to a green box, to a yellow box, to an orange box. Enzyme 5 finally converts it to a red box, which is the end product. Supposing this red box, this end product, was a protein that's involved in scab formation. You cut yourself and you need to form a scab. If this protein is involved in making scabs, then you're going to need a lot of it. So this pathway, starting with the blue box, is going to be running very fast to make a lot of this scab protein to help you heal. But once you heal, this red box, this protein, is no longer needed. But it's going to continue to accumulate because the pathway is still running. So the really neat part about this is that this first enzyme, enzyme 1, has a site that this protein can attach to and block this enzyme's activity. So when it does, when this accumulates, you no longer need the scab protein. It blocks this enzyme and shuts the whole pathway down. So that this end product will stop accumulating and in fact will all go away until you cut yourself again and you need more scab. So to clear how end product inhibition works, this is called both feedback inhibition and end product inhibition. It's a way of shutting down an entire pathway, and it's entirely controlled by the need for the end product.
Excuse me, teacher, it's a positive feedback or negative?
Positive or negative? Well, it's negative in the sense that it shuts the pathway down.
Because it's a stop the process or channel.
Yeah, it completely, if you have just a little excess end product, then you would not have complete inhibition of enzyme 1, and the pathway would be running slowly. So if you need only a little bit of the scab protein to finish healing your wound, the pathway would still run. But once it's completely healed, you don't need this product at all. Now it's going to be complete inhibition of the pathway. It stops. Anything else? Okay, that's another type of inhibition. And now we're going to talk about the energy part of it. Where does energy come from? Okay. And this is biological energy, the energy that we use to keep ourselves alive and do our activities. So there's a series of statements here that are all important, and they'll become meaningful as we go forward. But energy is harvested through electrons that are moved from one molecule to the next. And I'll show you this. Electrons carry energy, some of which will be transferred to intermediate molecules as electrons travel a downhill journey. The basis for this electron transfer is a series of reactions called redox, oxidation reduction reactions. I'll try to, well, some more statements. Substances attract electrons more than others. A substance that loses electrons is said to be oxidized. If it gains electrons, it's said to be reduced. And these two processes always occur in pairs. If oxidation occurs, there's simultaneously a reduction. So here is the simplest visualization of oxidation reduction. So on the left, you have compound or substances A and B. A is reduced. It has an electron. B is oxidized. Now, if A gives its electron to B, A is now oxidized and B is reduced. Now, Scott likes to use a mnemonic, which he calls oil rig, which you may find useful. That stands for oxidize is loss, reduced is gain. So oxidation is loss of an electron, reduction is gain of an electron. Now in biological oxidation, now here we're talking about an organic molecule, could be glucose, and we're talking about a coenzyme. Remember, coenzymes are involved in enzymatic activity, even though they're not proteins. NAD is an electron carrier. Now this reduced organic molecule has two hydrogen atoms. And you remember a hydrogen atom is a proton and an electron. It transfers these to this electron carrier called NAD, so that now the organic molecule is oxidized and the carrier is reduced. The carrier has a hydrogen atom and an electron and a proton. And this proton can carry energy. So this is what happens in an electron transport chain. There are many of these steps in an electron transport chain. But electrons can be moved through molecules called carriers. And the carrier molecules in photosynthesis are usually, they start with NADP. In respiration, there it's just NAD or NADH. So if you start with a reduced molecule, like glucose, for example, it can give extra electrons to this carrier, NAD, and make NADH. NADH in turn can hand off electrons to something else, for example, cytochrome, and cytochrome hands off to something else, and it goes on down the chain. And in the process of doing this, you see the little red ball here, energy is released. So what is NAD? Nicotinamide adenine dinucleotide phosphate. This all occurs with enzymes that are embedded in a membrane, which we'll talk about in a minute. And these enzyme complexes are huge, and I just put in, for example, NADH dehydrogenase, which removes hydrogen from NAD. It's a huge enzyme that has 45 polypeptide chains. So if you think of when we talked about tertiary structure and how one polypeptide chain is highly folded and cross-linked, think about how complex 45 polypeptide chains would be, all in one enzyme. So this shows the oxidized form of NAD, and the business end of the molecule is here. That can be reduced to NADH plus a proton, plus energy. And this occurs in a membrane. The electron transport chain occurs in a membrane. You can see that these proteins, these large protein complexes, are embedded in the membrane. And what they do is pass electrons from one to another in a stepwise process. And at each step, protons are released. And these protons accumulate on one side of the membrane. And you remember when you have ions accumulating on one side of the membrane, you now have an ion gradient. And if the ions are transported across the membrane, which you see here on the right, energy is produced. So at the final step here, 3 ADPs plus 3 inorganic phosphates give you 3 ATPs. And this is the process, this transfer of ions across a membrane to generate energy. That's called chemiosmosis. So you start with electrons with, say, an organic substrate like glucose, hands electrons off to NAD, to NADH, and you end up, the electrons go through this complicated pathway. The final electron acceptor is oxygen. That's why we breathe. We need oxygen to oxidize our food and generate energy. So it starts with NADH. The final electron acceptor is oxygen. And this I put in just to emphasize what chemiosmosis is, the electrons, and where it happens. So here's the membrane, and this could be either in the mitochondrion or the chloroplast. You have an intermembrane space, you have a matrix on the inside. Electrons start at one side, they go through various different steps within the membrane, and at the end you have chemiosmosis, where the electrons that have been accumulating on one side of the membrane now pass through it and generate ATP. And this is to show you spatially where all this is happening. So this blue object here is a cell. You can see it has various organelles. And this little orange thing is a mitochondrion. Could be a chloroplast, but in this case, it's a mitochondrion. If you look closely at that, what you see is it has an inner membrane, an outer membrane, a matrix, an intermembrane space. And where does chemiosmosis occur? It's on the inner mitochondrial membrane. It's this membrane that is highly convoluted on the inside of the mitochondrion. So that is where our energy comes from. So harvesting energy, the point here is that you take a molecule like glucose and oxygen, you could release all the energy in glucose at once. That would be an explosive release of energy, but that cannot be captured by cells. It all would be dissipated as heat. The cells would not benefit at all. You get water and carbon dioxide, but no energy. So what happens is the electrons are slowed down. Energy is released very slowly. You start with glucose, and that's split to carbon dioxide and water way down here at the end. And you think of it, you can think of it as a series of stair steps, because the energy level is going down as electrons travel from one carrier to the next. So A, energy input, splits glucose into carbon dioxide, electrons, and hydrogen. B, the electrons go down this pathway, and you remember what this really is. Every step is this. At every step, you're transferring to an electron carrier and then to another carrier. And then energy is released as the electrons are harnessed for work, and the electrons and protons and oxygen combine to form water. So the end product of glucose is carbon dioxide and water. And remember, photosynthesis is the opposite of that. Carbon dioxide and water plus sunlight makes glucose. All right, and we're going to talk a lot more about how ATP is made in future slide sets. But this is the basis of it. There are three possible ways to make ATP out of ADP. One is called substrate level phosphorylation. That's a direct enzymatic transfer of phosphate from a phosphorylated molecule to ADP in order to make ATP. A second way to produce ATP is called oxidative phosphorylation. In this case, electrons from an organic compound like glucose are passed through a series of reactions, which we've just been talking about, the electron transport chain, to inorganic molecules like oxygen. And at the end of this, a process known as chemiosmosis generates ATP. And finally, there's photophosphorylation, where the pigments of photosynthetic cells, different types of chlorophyll, trap light energy and convert it to chemical energy. And the way they do this is through electron transport through this chain that we've just been talking about to chemiosmosis. So you can see that photophosphorylation and oxidative phosphorylation are very similar. They have different starting points. So oxidative phosphorylation starts with glucose. And you end up generating ATP from an energetic molecule like glucose. Photophosphorylation starts with light, whereas light splits water, and along with carbon dioxide, you make glucose. And then this is just a summary to illustrate these three different ways of making ATP. And we'll talk in more detail about these in the future, but for right now, what you can remember is that substrate level phosphorylation is one simple step. There's no electron transport chain involved here. And you start with one ADP and you get one ATP. Oxidative phosphorylation involves an electron transport chain where you start with glucose, which is split into two molecules called pyruvic acid, pyruvate. Then this is made into acetyl-CoA, and it goes into, we'll talk about what a Krebs cycle is later. But as this goes on, you're generating carriers, NAD plus protons, NAD plus protons. So by the time you get to the bottom here, you have 10 NADHs, 2, this is flavin adenine dinucleotide reduced, 2 FADHs, so that, and these are all releasing electrons into the electron transport chain, so that if you start then with one molecule of glucose, you end up with 38 molecules of ATP. Now contrast that with substrate level phosphorylation, where you start with 1 or 2 ADPs and end up with 1 or 2 ATPs. That's what you see in this first step here, 2 ATPs from substrate level phosphorylation. All the rest of these are generated through electron transport chains. So oxidative phosphorylation is much more productive because of this slowing down of the electrons than substrate level. And as I said, photophosphorylation is much the same as oxidative phosphorylation, except that you start with chlorophyll and light. Light provides the energy to excite electrons, split water, and the electrons are then transferred through an electron transport chain in the membrane, so that in the end, ATP is generated from ADP. And I put this little diagram, I showed you a very complicated diagram of what chemiosmosis is. Here is a very simplified way to visualize it. An ion gradient has potential energy. Remember potential versus kinetic? So an ion gradient is potential. That is, there are ions accumulating on one side of the membrane. When those ions go through the membrane, that is kinetic energy. So chemiosmosis, the simple way to state it, is the movement of ions across a semi-permeable membrane down their electrochemical gradient, generating ATP as they do it. So you see a high concentration of ions on one side, a low concentration on the other. When the ions pass through, and these are now electrons, when they pass through, ATP is produced, and that's called chemiosmosis. I think that's the last slide. Yes, it is. So I tried to go through this slowly. I know that it's a lot of information. It's complicated, and some of it will be elaborated further when we talk about specifically photosynthesis and specifically respiration. But any questions about anything so far?
So I have a quick question on the, I forget exactly what it was, like the cyclical inhibition.
Oh, feedback inhibition?
Yeah. So in a way, is that sort of like how in ethanol fermentation, the yeast create the alcohol until eventually it gets to the point where the alcohol level is so high it kills off the yeast, therefore sort of ending the process?
I don't know that that's a perfect analogy. It certainly, the end product certainly does stop the reaction, but I think in this case it's because alcohol is toxic to the yeast. It's not necessarily binding to an enzyme and shutting it down.
Right. I was speaking more like the most general way possible, like just a rough overview of it.
Conceptually, it's the same in that you have an end product accumulating that stops the reaction. It stops the accumulation.
All right. Thank you.
But keep in mind, it's mechanistically, it's not the same.
Oh, yeah. Completely different. Just the general way of thinking about it. Okay. Thank you.
Anything else?
I'm not sure if you said this earlier, but I know you were talking about it. Did you say when the test date was for our next day gonna be? Like the when's gonna be?
Yes. Test 3 is a week from today, Monday the 6th.
All right, perfect. Thank you so much.
Okay. Anything else?
I have a question from the previous slide for enzyme.
Okay. Which one?
The previous one, I think it's 35.
Let's see. Okay, here's feedback. So which way do I go from here?
Back.
Back?
Yes. So that's one.
This one?
Yeah, this one. So why the enzyme is changing its shape? So
Why
what's
does
the reason? What's the reason that the enzyme is changing the shape?
Yeah, you remember that this enzyme, it's pictured here as a blob, but it really isn't a blob. Let's see. Where's Tina? Yeah. So this illustrates an active enzyme, which is highly folded and highly cross-linked. So it's not a blob. And so somewhere in here is an active site for a substrate. If you have non-competitive inhibition, there's another site somewhere where the non-competitive inhibitor can bind. And when it does, it changes the configuration of this entire complex so that the active site over here is no longer available. It's changed shape because of the inhibitor over here. Now, in the case of competitive inhibition, the substrate and the inhibitor are competing for the same active site in some part of the molecule. In non-competitive inhibition, they're not competing for the same site. They have two different sites, but the site for the inhibitor changes the shape of the active site. Is that clear?
Yeah, but I'm saying that it's like the instruction, or there is an issue that's make it change, or it's. I mean, like if there's a disease or it's a normal instruction in the body, sometimes you need an enzyme to work, sometimes you don't need the enzyme to work. So it will change the shape, so it will not fit. So there is no active, it will be not active.
Yeah.
So so it's normal.
It's not a result of a disease. It's all perfectly normal, and it's because you can't have enzymes running full speed all the time.
Oh, that's what I was I
So,
get that.
so if you think about, we have thousands of enzymes in our bodies. Most of those enzymes at any point in time are not active. They're being shut down by one mechanism or another.
That's why my question. Yeah, I just want to know they do that, so they shut down the enzyme because they don't want them to work right at this point.
Right.
So that's
I tried to make the analogy with feedback inhibition of wound healing. So if you have a wound that needs a scab, there are a lot of proteins that go into building that scab. And if the product of a pathway makes a scab, where is that? I'm going the wrong way. Where is it? So if this end product is needed for, say, wound healing, then as soon as a protein is made, it's scooped off and transferred to the wound to help in wound healing. But as soon as the wound is healed, this product isn't needed anymore. But yet the pathway is still running. There has to be a way to shut it down.
to stop that, we will change the enzyme shape, so that's what will done.
Right. So when this end product now, it has a binding site in the first enzyme in the pathway. And when it binds to that site, it changes the shape of this enzyme, so it's no longer active.
Yeah, I got it.
No longer converts substrate to product.
Yeah,
so
clear.
And if this first step doesn't happen, none of them happen, and the whole pathway shuts down.
Sounds good. Thank you.
Professor, I have a question. This is Mary Kate. For the worksheet enzymes and catalysis, the number 10 and 11, I don't know, or I don't understand what do you want specific for these questions.
I don't have the worksheet up right now. Let's see. Okay, we're out of time, so anybody who wants to leave can. I'm going to stop sharing and try to find. This is the worksheet.
Sorry.
I'll try to find it. See where,
Okay.
I think that's enzyme CD. Now you said questions 10 and 11.
Yes.
Okay. 2 types of chemical reaction.
Yes.
Require energy. There, and what I'm looking for here is endergonic and exergonic.
Okay. Thank you.
I
Okay.
do have a quick question on the worksheet actually for number 12.
Okay.
Are you looking for specific examples or just general like size of molecules?
Let me get that back up there again.
I'm trying to be vague.
Oh, for number 12?
Yeah.
Yeah, I had in mind general. So catabolic pathways release energy. They break down large molecules into small ones.
Okay, cool. I had simple or complex into simple, but okay.
It's the same thing. I'd accept either of those.
Okay, sounds good. Thank you.
Okay, anything else before we go?
Have a good day.
All right. See you all on Wednesday.
See you on Wednesday.